back to the NPTEL series of lectures on animal physiology. So, we are in the section of respiration. So, we started with the first lecture of respiration, where we talked about the structure of the lungs and how the trachea, which is a cartilaginous structure, splits up into bronchioles, and from the bronchioles to alveoli, and how the exchange takes place at the alveolar surface. That was the first part, and we highlighted the fact that it is the partial pressure difference of oxygen and carbon dioxide which results in the exchange of gases. In other words, what is exactly happening is that the blood which is enriched in carbon dioxide releases its carbon dioxide and picks up the oxygen which we inhale, and this whole process falls under the respiration. So, in the last class I told you that in this section we will be talking about two aspects. One aspect is how we calculate the partial pressure and how we have the complete table of the partial pressures, and we could see how this from higher partial pressure, how it goes to lower partial pressure, especially in terms of oxygen and carbon dioxide. And then what we will do, we will talk about how the blood and carbon dioxide is being carried, how the oxygen and carbon dioxide is being carried in the blood, what are the different mechanisms blood employs to carry this two very important gases which are involved in the respiration as well as metabolism and pretty much everything in our body, and most importantly the buffering. And in the third part we will be talking about the buffering mechanism which the blood follows in order to ensure that we do not suffer from any form of acidosis or alkalosis, but as many people have heard or see, it is the extreme acid conditions which even lead to conditions like cancer and many other things. So do alkalosis. Okay. So, we have to always maintain a very stable or kind of a very pH range cannot vary significantly from 7.2 to 7.3. We are slightly on the acid alkaline side, but that is the permissible limit, other than certain organs in our body like a stomach, what we have discussed in digestion, which is extremely low pH. Other than that, most of the body, rest of the body functions at a stable pH around 7.4, 7.3 likewise. Okay. So, let us start with, and as I told you in the last class, please go through the Dalton's law of partial pressure. So, let us come back to section 7. This is the second lecture. So, if you put the partial pressure, so there are three gases we are dealing with, okay? Oxygen, carbon dioxide, and nitrogen, which are present in the environment, and of course, significant amount of water vapor in the, and that also exerts its partial pressure. Okay, partial pressure which we indicated by say Pb. So, Pb is equal to partial. So, P stands for partial pressure. Okay, partial pressure of nitrogen plus partial pressure of oxygen plus partial pressure of partial pressure of carbon dioxide and partial pressure of water vapor. So, these are the four components which constitute the complete total partial pressure equation. Okay. So, now if you know at least one component, and if you know, so always remember this partial pressure is a function of the altitude where you are. Okay. So, if you are at the sea level, so that is where it is being measured. If you are at the sea level, the partial pressure is mostly at atmospheric pressure is 760 millimeter mercury, but if you go up in the mountains, then the partial pressure, the atmospheric pressure decreases, because for the simple reason, because all these gases what you see, their concentration reduces as you go up. Okay. So, as the concentration decreases, so do the partial pressure. Okay. So, at different pressure zone, if you are at 760 millimeter mercury, the pressure is different. If you are at a higher altitude, pressure is different, and as you go down inside the ocean, then the pressure is different, it goes up. Okay. So, these are the basic fundamentals which all of you must have studied at some point or other, class 6, class 7 or somewhere or other. It is just for you guys to rehash and keep that in mind whenever. So, what we will be doing here, while we will be doing the calculation, we will be doing the calculation based on at the at the sea level, assuming at sea level 760 millimeter mercury. Okay. So, at 760 millimeter mercury, let's get back. So, when our assumption is very clear. So, when the barometric pressure at sea level, I am just putting at SL at is 760 millimeter mercury. Okay. And partial pressure of water, I just put PP, sorry, since I am talking like that. So, the partial pressure of water vapor is 747 millimeter mercury. This value, I give you this value. So, under this value, if I ask you that calculate the, one second, again there is a confusion here. At this stage, I ask you, please calculate the partial pressure of oxygen. What will you do? Okay. This is what I am asking you to do. So, okay. So, we know the oxygen concentration in air is 20.8%. Okay. This another value is known to you, and the total partial pressure is denoted by Pb. Okay. So, what we do? We know one of the values, okay, that is 47, which is contributed by water. Okay. So, PO2, how to calculate PO2? That will be 20.8% of 760 minus 47, and the 760 is coming added together at the sea level. So, if you really solve this whole thing, 20.8%, and this becomes 3, 426, 723, and if you totally calculate it, it comes out to be 100 millimeter mercury. So, this is the contribution of oxygen. Okay. If this is the contribution of oxygen, what is the contribution of carbon dioxide, PCO2? So, for that you need to know what is the concentration of CO2 in the air, which is 0.03, which is very negligible. Okay. So, if you have to calculate, then Pb again it is the same thing, Pb minus 47, which is your contribution from water. Okay. And then what your PCO2 becomes 0.03%, 760 minus 47, 0.03%, and this value was how much you calculated? Was 47. So, 3, 4, 5, 713. Okay. So, that is fairly negligible, approximately almost 0, okay, if you calculate it. So, basically in the air situation is like this. In the air, your CO2 contribution is fairly low. This is the regular air, what, and oxygen we have talked about, the partial pressure of oxygen in the air is around 150 millimeter mercury. Okay. And at this point, we are only concerned with these two gases, because those are the ones which will help us. So, if you really form the table, so this is what I was telling you. So, partial pressure of oxygen and CO2, we do the calculation at two different level. Okay. So, this is PO2 in millimeter mercury, and this is PCO2 at millimeter mercury, and this is for the inspired air, the air which you are inspiring, inspired air. We have calculated this 150, and this is approximately 0, whereas alveolar gas, as well as I am just putting both the arterial blood, which is the blood which is laden in carbon dioxide, which is coming to the lungs for purification, both of them. This is around 100, and whereas this is around 40. So, let us consider this as a common thing. This is pretty much the same. Okay. Now, what will happen? So, now what essentially what happens is that here you see partial pressure is higher. So, automatically oxygen gets into the arterial blood, whereas here if you look at it, partial pressure of carbon dioxide is much more higher as compared to the outside air. So, the carbon dioxide moves like this. This is essentially, this is one of the most key thing what you have to kind of realize, that this is how this whole exchange takes place because of this partial pressure. Use the simple logics. So, what I expect the take home message for this particular part will be, you have to have a very simple clear cut understanding of partial pressure and atmospheric pressure. If you know the atmospheric pressure, if it is at 760 millimeter mercury, I did the calculation. But if I tell you that you are in a very high altitude, say Machu Picchu in Peru, or somewhere in Ladakh or Leh, or somewhere in Himalayas or in Andes, how are you going to calculate? So, what you need to know is what is the atmospheric barometric pressure, and if you know the barometric pressure and if you have these values reasonably close to it, you can make the calculation. Okay. So, these are the simple ways by which you can make all these calculations, which regulates your physiology. As we will go through, you will see it. Okay. So, now what we will be talking about after this is how oxygen is carried in the blood. So, if you remember in the last class, I told you, in spite of the fact that oxygen has a low molecular weight as compared to carbon dioxide diffuses much more higher as compared to oxygen. The reason being, carbon dioxide is very readily soluble, whereas in the case of oxygen it is not readily soluble. So, that poses a problem for oxygen to be carried. So, oxygen needs a special treatment to be carried all along your body, because it really cannot diffuse into the plasma so readily, or even if it diffuses at a very low concentration, which is not good enough for metabolism to take place. So, how the body handle that kind of situation? The way the body handles is this: body has a specific oxygen carrier, and they are found in the red blood cells. So, whenever you go to a doctor asks you, when they do a blood analysis, they ask you what is the hemoglobin percentage. Okay. Generally, it should be around 12 to 14, okay. But sometime it falls down to 7 or 8 or to 4 sometime, even worst case. Okay. That means that there is some problem, because your body is not carrying sufficient oxygen. So, if the body does not carry sufficient oxygen, what essentially that leads to is that your metabolism is compromised, your nervous system functioning is being compromised, and these are some of the natural phenomena which happens in the highlanders at high altitude. Very suddenly you go to a high altitude, oxygen is low. So, automatically if the oxygen is low, the hemoglobin is not completely binding to the oxygen. So, these are the kind of situations. So, let us get into it, and that will help you to realize, and there will be some stark differences you will come across, how oxygen is being transported and how carbon dioxide is being transported. Okay. So, let us start with oxygen carrier in blood. Okay. So, here you have the red blood cells, biconcave disc. Okay. And this I have already mentioned, it has a poor solubility in plasma, and this is an RBC, and within RBC you have these hemoglobin molecules sitting there, just putting them at HB, hemoglobin. This is a protein, and we will talk about the structure of this protein, okay, and which is your oxygen carrier. So, if you look at the hemoglobin molecule, so hemoglobin molecule is something like this. It has 4 subunits like this, and within the subunit it has a interesting center. Come to that. This is a single molecule of hemoglobin. So, these two chains, this and this one, is called alpha chain. These are the proteins, okay, and these two chains are called beta chains. Okay. And this thing, this red color thing, is basically iron porphyrin. This is the one which binds to oxygen. So, that is why you will see whenever there is a deficiency of hemoglobin, people say you take iron rich diet, or if you have to increase your blood, they say colloquially they will say you take iron rich diet. So, this is where the iron plays the role, and I request you people, please go online and check what is the structure of porphyrin. Porphyrin is basically, I will not give you the full structure, just look into it. It will be something like you will come across has something like a structure you will come across like this, and I want you guys to really go there, go online and check how all the bondings are taking place. It will be a structure like this. You will have the iron sitting here. Okay. And one more thing I request you to look at, what is the oxidation state of iron here? Is it +2 or is it +3? Is it ferrous or is it ferric? Please look this. This is the exercise I expect you people to do. And there is another closely molecule to porphyrin, where iron is being replaced by magnesium, and that is what is called chlorophyll. They are very close to each other, and chlorophyll, as you know, is the one which helps to trap the sunlight. Okay. So, they are very close molecules, and look into it. I expect you guys to look into this structure. So, this is basically the overall skeleton of porphyrin, and go through it and look for the oxidation state of iron, in what oxidation state of iron stays as porphyrin, and this is where, this is the site where the oxygen is getting bound. Okay. So, I will not go into the technical details of it in depth, because this is not, these are covered in biochemistry, but I expect you guys will enjoy if you go through this. Okay. So, there are basically in one molecule, if I had to say for example, one hemoglobin molecule like this, one hemoglobin molecule could bind to, as you, if you remember in the first diagram I was doing, 2 beta chain, 2 alpha chain, it is a site to bind to 4 oxygen molecule. So, if I put the blue as oxygen, likewise. Okay. So, this is that red zone, oh sorry, the red zone which I was drawing in one of the previous slides. Okay. So, 4 oxygen bind, this is called oxyhemoglobin, which is laden in oxygen. Okay. So, basically oxygen binds very loosely out there, because it cannot bind very harsh, because if it binds very harsh, then it will not be able to dissociate. So, basically what is happening is that B4 plus O2, 4 I put because there are 4 binding site. Essentially what is happening is that Hb4 plus O8, because O2 multiplied by 4, that makes it O8, is Hb4 coming. So, this is basically how the oxygen is binding, and based on the saturation level of oxygen, you can term it as, likewise, if there is only if all the 4 sites are occupied by oxygen, this is 100% saturated. If 1, 2 are, it is 50%. If only 1 is, then it is 25%. If none, this is unsaturated. And based on this, you can draw the saturation, which is also called oxyhemoglobin dissociation curve. Okay. Oxy, one second. Oxyhemoglobin dissociation curve. Okay. This is basically what is essentially says this. On y axis, you have hemoglobin saturation. I was telling you hemoglobin saturation. So, it could be the 25%, 50%, 75%, 100%. Okay. And whereas on the x axis, you have oxygen tension in millimeter mercury. So, this is at a 50, 100, 150 likewise. And if you draw the graph, it shows almost an S-shaped curve here. Okay. And if you look at it somewhere here, it will be the venous blood, which has lower saturation of oxygen. As you know that most of the oxygen is being used up by the tissue. And if you look somewhere out here, this is where you have the arterial blood, which is filled with oxygen, except the artery which is taking the carbon dioxide rich blood to the lungs. Okay. And you can see the difference in the oxygen in the hemoglobin saturation of oxygen. And at this part of the curve, this is almost independent of oxygen concentration, because as long as it has the binding sites, all the hemoglobin molecule has a limited number of binding sites. It has only 4 binding sites. Okay. So, assume a situation when all the hemoglobin in your blood, in your red blood cells, and plus all the red blood cells are completely saturated with oxygen. So, you do not have any other part where oxygen can bind, and as we have already said, the oxygen dissolving power, solubility is very, very low. So, automatically there is no room for oxygen really to bind. So, under that situation, this curve reaches a peak. What you see a plateau, the plateau is reached because there is, even if you even if you add oxygen at that point of time, there is no point, because there is no more oxygen binding site left in the hemoglobin, because they are all filled. So, when the plateau reaches, it is free from any oxygen concentration. Okay, so, remember that, this is very important to understand. Okay, coming back to where we were. Okay, so, this is the zone which I am trying to highlight, that this is completely free from oxygen. Okay. And another thing what I wanted to highlight here is that at this zone, if you look at it, so what essentially is happening is, what is happening here when the arterial blood is coming, arterial blood and the venous blood. Okay. This blood is coming with very high oxygen, and then when it goes to the tissue, if this is the tissue, just assume this box as a tissue, it gives away the oxygen and picks up the carbon dioxide. It is here, you can figure out, if you see the saturation zone, that is what happens. The oxygen tension is lesser out here, and if you see the previous graph, it was exactly like that. And this oxygen tension changes further during 2 different kind of, whether you are at rest or whether you are at exercise. At rest, your oxygen consumption is low oxygen consumption, whereas during exercise, oxygen consumption is much more higher. So, automatically the saturation goes much more faster. Okay. So, remember that at the lower part of the graph, where basically the venous blood is having a lower oxygen tension. So, see that graph, and that graph is reached very fast when you are doing a lot of exercise. Okay. So, this is something which I expect you people to understand. Okay. From here I will move on to the next slide, where basically some of the facts what I expect you guys to understand is that there is 15 gram hemoglobin is present in 100 ml of blood. This will help you in all kind of calculations. So, this is basically what we talk about, how much hemoglobin is present, and that each hemoglobin can carry 1.34 ml of oxygen. This is very important, and the oxygen content of arterial blood at 100% saturation is 20 ml oxygen per 100 ml of blood. These are some bits and pieces of information which will help you to do any kind of calculation. So, that is why I wish to highlight that this is very important for all the calculation purpose. If you know these values, it will be fairly easy to do any kind of calculations. Okay. After the oxygen, so we talked about the hemoglobin and talked about the porphyrin structure of hemoglobin. Let us talk about how carbon dioxide is being carried in the blood. So, in case of carbon dioxide, there are three different mechanisms. There is no specific, as such there is no specific carrier. That of course, hemoglobin does bind to carbon dioxide and it called carboxyhemoglobin, just like the oxyhemoglobin when oxygen binds to the hemoglobin. There is also whenever oxygen is being thrown out and carbon dioxide binds to it, that is called carboxyhemoglobin. So, apart from carboxyhemoglobin, there are two other routes by which carbon dioxide is being carried. So, what we will do now, we will enumerate all the different routes by which carbon dioxide is being carried out, is being carried all over the body. Okay. So, let us get back to the carbon dioxide carriage by blood. Okay. And this is very readily soluble in the blood, as I have told you. So, first is as and I will discuss all of them separately, as a bicarbonate, as a bicarbonate ion, which is HCO3 minus in plasma. This is one route, the route 1. Route 2, carboamino compound, which is formed when CO2 and NH3 group of protein, which is essentially we are talking about carboxyhemoglobin. Okay. There is a third way of carrying, which is as a dissolved molecule. This is the easiest because it has the highest solubility. I have already mentioned that these are the three different ways by which carbon dioxide can be transported. So, how it does in the bicarbonate, because that has another significance in pH, that is why I will come back to that. When CO2 plus H2O, when these two mix, there is an enzyme which is present in the body which is called carbonic anhydrase. Carbonic anhydrase. Okay. What carbonic anhydrase does is that transform it into H2CO3. To go to the next page to complete the reaction, then H2CO3 basically has the ability to dissociate H plus HCO3 minus. Okay. So, if I put the whole reaction in perspective, then this is like this: CO2 plus H2O, reversible reaction. This is where Ca, carbonic anhydrase, comes into play, H2CO3, and then from here it breaks down to H plus HCO3 minus. Okay. And this carbonic anhydrase is present in only in the RBCs. It is not present in plasma. Mark my word, not present in plasma. So, this reaction has to take place inside the RBCs. Okay. And if you have to do a complete outline that which of these component is contributing how much for carbon dioxide travel, then we will see. So, let us enumerate them. One is the bicarbonate we talked about, bicarbonate, carbamino compounds, and then you have dissolved CO2. So, this is 10% dissolved one. Carboamino compounds, that is carboxyhemoglobin, likewise is 30%, and the major chunk is in the form of bicarbonate, and this is very important that bicarbonate is the one which carries the maximum amount of it. Now, from here what I will come to the concept of pH, how pH is being regulated. So, this is a very important fact you have to realize the pH. So, in the beginning of the lecture I told you that pH is exceptionally essential in terms of maintaining the proper homeostasis of the body. So, the pH is, if you go back to that reaction, if you remember the reaction, it is just I draw carbon dioxide plus water making HCO3, and then HCO3 is H2CO3, H2CO3 is dissociating into H plus and HCO3 minus. This reaction is one of the most fundamental reaction, and depending on forward reaction, backward reaction, rate of forward reaction, rate of backward reaction, because you have two control zones. This is the one which helps to maintain the pH, and that is what we are going to deal now, how this particular reaction helps to maintain the pH of the body. Okay, coming back to the sheet. So, the pH of the arterial blood is held constant by the, so this is the one which is helping in the whole buffering by the buffering action of HCO3 minus and CO2. This is very important, CO2. This is a system which helps in all our buffering, and by blood protein, which is mostly blood protein which is involved in, this is the hemoglobin. This is something which you have to realize, and this could be stated by the Henderson-Hasselbalch equation, and please go through this, those of you have forgotten it. Okay. Basically, using Henderson-Hasselbalch equation, we can formulate the pH of the blood can be calculated by adding a constant, so remember this, adding a constant term to a variable term given by the logarithm of the ratio of the HCO3 minus concentration to CO2 concentration. And if I put the mathematical equation here, that will be pH is equal to 6.1 plus log of HCO3 minus molar concentration divided by concentration of CO2. This is very, very important. Okay? And if you see that, if we say the value of log 20, which is equal to 1.3, the pH of blood at that situation will be 7.4. So, in order to have a 7.4, what will be the situation is just back. Okay. So, whenever HCO3 minus upon CO2 is 20, that is 6.1 plus log 20, which is equal to 6.1 plus 1.3 makes it 7.4. So, what are the implications? So, if you look at it, so these values, if you know the Henderson-Hasselbalch equation right, that pH is equal to 6.1 plus log of HCO3 and CO2 upon CO2, then you can make this whole calculation without any problem. And this is exceptionally essential. And what are the implications of Henderson? So, let us talk about the implications of it. Well, I am just putting HH, Henderson-Hasselbalch equation. Okay. the pH of the blood can be maintained at around normal level of 7.4 by balancing operation of renal system, which is your kidney system, okay, which actually regulates, which as you come to the kidney you realize this, which actually regulates is HCO3 minus concentration and the respiratory system, which regulates your carbon dioxide concentration. So, this is exceptionally important for you people to realize that these are the two situations which regulates the pH. So, you have an interplay of kidney or the renal system and respiratory system. So, this is regulating CO2, this is regulating HCO3 minus. As long as these are properly balanced, your pH in the body will be balanced. Okay. Another thing for you to important to remember is that pH of the blood will, as this is just the direct implication of it, will deviate from 7.4, which is the normal value, whenever your HCO3 minus is to CO2 ratio of 20:1 shifts either upward or downward. If, say for example, if your HCO3 minus is to CO2 ratio is more than 20:1, then we are hitting on to situation called alkalosis. It means the body is alkaline. Whereas, if HCO3 and CO2 ratio is less than 20:1, then we are hitting into acidosis. So, this is what you have to remember very clearly, and there are a couple of more terms which are very essential. Acidosis could be because of, this is basically higher level of CO2 and lower level of HCO3, which is basically called metabolic acidosis, and this is called respiratory acidosis. Okay. Because for the simple reason that CO2 percentage is governed by the carbon dioxide. So, whenever CO2 percentage goes down or goes up, it is at the respiratory acidosis or alkalosis. Whereas, in the case of HCO3 concentration, which is regulated by the kidney, it is basically the metabolic acidosis or metabolic alkalosis. So, these are the aspects which we covered today. We talked about the partial pressure of carbon dioxide and oxygen in the air which we inhale, and the level of carbon dioxide and oxygen in the blood. That we talked about the carriers of oxygen, carriers of carbon dioxide, and then we talked about how this CO2 HCO3 buffer system helps to maintain the pH of the body. So, these are the overall things, and kindly go through the notes which I have given you here, and that will help you to understand, and always be very clear about your basics. Okay. Thanks a lot.